So we're lucky today to have a great friend of Alex's presenting this lecture today, Dr. Will Parsons, who's a longtime friend and was a grantee, I think, as a young investigator and then an early A awardee. Dr. Will Parsons is a board-certified pediatric oncologist, and he is a deputy director of Texas Children's Cancer Center and Hematology, Texas Cancer and Hematology Center down at Baylor College of Medicine in Houston. When we met Dr. Parsons, he was at Johns Hopkins, I think, finishing up his fellowship or his postdoc, I can't remember which. Fast forward to today, Will has been a longtime member of the scientific advisory board for Alex's, so he helps us determine where we want to go with our grant funding, and has been a great scientific advisory board member. I think one of the other highlights for Dr. Parsons is heading up the NCI-COG pediatric match trial, which is the first nationwide precision oncology trial for children with relapsed and refractory solid tumors, lymphomas, histiocytosis, and as a steering committee member for the NIH Pediatric Early Phase Clinical Trial Network. So Dr. Parsons is very into translational and clinical medicine. With that, I'm going to turn it over. We appreciate you being here today, and thank you for everything you've done for Alex's over the years.
Thank you. I really appreciate it, Jay, and I'm very grateful for everything that Alex has done for me and my research program and our patients over the years. So it's been actually a wonderful opportunity to work with you. So I am going to bring up my slides here. And then let's see, view. How do I get to view slideshow? Does that look good? Is that the correct format?
Yeah, that looks good, Will.
Awesome. Thank you. I appreciate it. I just am notorious for screwing this up. So it's very exciting to have the opportunity to talk here today. I'm going to be speaking about my experience and our collective experience in the area of pediatric cancer genetics and genomics. It's really been a remarkable period from when I actually started training back in the early 2000s. I first received funding, as Jay mentioned, from Alex's Lemonade Foundation for Young Investigator Award back when I was a fellow in 2007. So it's been solid 15 years and counting of relationship with the foundation. So I'm going to be talking about what's commonly referred to as precision cancer medicine. And on the screen here, you can see one definition of this from the NCI. And, excuse me, I'll just read it because I think it's important. It says, a form of medicine that uses information about a person's own genes or proteins to prevent, diagnose, or treat disease. In cancer, precision medicine uses specific information about a person's tumor to help make a diagnosis, plan treatment, find out while treatment is working, or make a prognosis, also called personalized medicine. So a couple of comments on this. One is, I think as pediatric oncologists, we consider that we're practicing personalized medicine or precision medicine, in every aspect of how we take care of patients. So this is really just talking specifically about using genetics for that purpose. And the other is, as I think, hope I'll make the case over the course of my talk, over 20 years, precision cancer medicine in this context, meaning the era of us being able to do essentially a genome-wide analysis of a tumor or of a patient, has really become possible and then become quickly transitioned over that time period to being a fundamental part of how we take care of patients. So in some ways, I think it's actually pretty remarkable that what we would talk about as precision cancer medicine as a field has been invented and then almost been eliminated isn't the correct word, but incorporated into standard care over that time period. So it's been pretty remarkable progress. For me, the start of this was this paper I read back in 2002 from the group at the Sanger Institute in the UK. Basically, where they were looking at one specific gene, the BRAF gene, and sequencing it and identifying mutations and characterizing how these were found not just in melanoma and a few of the different tumor types where that was already known, but in some fraction of patients in a large number of diseases. And for me, really just opened up the possibility in our thinking of trying to figure out how to treat cancer, not so much in a histologic diagnosis specific way, but rather identifying molecular or genetic vulnerabilities that cross tumor types. And so over the next handful of years, large-scale genomics became a possibility. So the first exome sequencing studies, the first whole genome sequencing studies. I'm highlighting this one just because it's one that I happen to have slides for from being involved in my time back in the lab at Johns Hopkins. But really for the first time, using sequencing to, in an unbiased genome-wide way, to identify molecular vulnerabilities, such as this discovery of the isocitrate dehydrogenase 1 and 2 gene mutations in adult gliomas, and also a significant fraction of adolescent and young adult patients in particular with high-grade gliomas. An example of a case where as a gene, we had no knowledge of it being connected to cancer in any way before the study started. And after the study was completed, opened up a new realm of kind of investigating the role of these genes in cancer metabolism more broadly for brain tumors. And then over the next handful of years, these research sequencing studies just continue to explode. And I'm just highlighting a couple of them, but this is an example of early characterization of high-grade glioma in children. The previous slide had been primarily and exclusively in adult patients. And it's, the details are unimportant, as likely everyone who's listening has seen these types of oncoprint or these types of graphs before. Each column is a patient, each row is a clinical characteristic or a particular gene. And so this is one just emphasizing in these pediatric high-grade gliomas, including DIPG, finding for the first time the characteristic alterations in these histone H3 genes in these tumor types is one of the defining features of their biology. And again, a case where these genes were unknown to be relevant to cancer in any type before this. So really just opening up the possibility of new avenues for better understanding and treating pediatric cancers. Fast forward another several years, and there are genome scale studies being conducted by investigators both here in the US and in Europe and elsewhere, characterizing essentially all of the common adult and pediatric cancer types. This slide is one that's commonly shown, just showing the mutation rate. So the y-axis is the number of mutations per tumor, and the different categories you can see are the different cancer types. And one point that's often made is that pediatric cancers have fewer mutations than adult cancers, which is definitely true. But the key point is there are also some pediatric cancers, such as the hypermutated high-grade gliomas and some of the others where there are many, many mutations, which is relevant for both thinking about targeted therapies as well as checkpoint inhibition and other treatment strategies. But then also, there are a significant number still, even though there are fewer mutations of all types in these pediatric tumors, there are key gene drivers that have been identified for each of them. But again, over a kind of a period of close to a decade from the first exome or genome-wide sequencing studies, really in-depth characterization of all these tumor types. And so then in parallel, this precision medicine strategy has been incorporated into every aspect of how we take care of our patients in pediatric oncology. So up until now, I've been talking about exclusively the research sequencing studies. But here we're looking at for example, as any of the folks who take care of patients with brain tumors in the eyes will be familiar, a snapshot from the WHO most recent 2021 classification of CNS tumors. We're starting for the first time, the molecular features are being utilized in addition to just the histologic description or what the tumor types look under the microscope. You can see this for any of these categories, from the medulloblastomas, which now have all of these molecularly defined categories: Wnt activated, sonic hedgehog activated, and p53 wild type, sonic hedgehog activated, and p53 mutant, the various ependymal tumors. Suddenly, I mean, not suddenly, but in relatively short order, a number of these genes and molecular groups that were not known 10 to 20 years earlier are a fundamental part of how patients are being diagnosed and treated, and form the basis of how we think about these cancer types. Even more broadly, or more expansively, methods such as methylation profiling, and this is as likely all of you have also seen a map showing Tisney, what's called Tisney plot, and with each of these different clusters and each of the different colors being a different molecular class of brain tumors, so consisting of both pediatric and adult cancer types, and really making the point that there's incredible diversity in terms of the molecular features of these tumors that can be picked apart using methods such as methylation profiling or RNA sequencing to understand the different tumor types as well as the different tumor subtypes, but really making it clear that we need to be thinking about these cancers not just in terms of what they look like under the microscope, but rather what their molecular features are and what this might mean for a better understanding their biology in cancer. And so this is, through various initiatives, something that's now being utilized clinically. I wouldn't say as a routine test, but being used increasingly commonly over the last several years. So beyond diagnosis, and for all of these slides, I'm focusing pretty largely on solid tumors, clearly in a very equivalent talk with perhaps even more examples can be provided for leukemias or some of the hematologic malignancies. But here, this is a slide, for example, I think just making a point of the capabilities of genomics to provide improved molecular prognostication and risk stratification, which is key clearly for the design of future clinical trials and how we treat these patients. So this case, for example, these are the 4 primary subgroups of medulloblastoma. In the upper left corner, the Wnt subtype, then on the upper right corner, sonic hedgehog, and then group 3 and group 4, showing for a particular trial what the observations were for the survival probability, event-free survival probability over the course of this trial. And you can see it's relatively long follow-up over 10, 11 years. A couple of key points, one being that there are quite different prognoses for these different molecular subgroups, including the Wnt pathway tumors in the upper left, where the prognosis is really quite remarkably good, relatively speaking, and then others such as the group 3 tumors where it's less impressive. But then also that the distinctions between, for example, one of the study questions in this particular trial was whether the addition of carboplatin or lack of carboplatin would have an impact on the prognosis of the patients. And you can see for some of these subtypes, such as the group 4, virtually no difference at all between the orange line and the gray to black line. Whereas others, such as the group 3 tumors, there did seem to be some significant difference. So different subgroups having different molecular features and also different responses to therapies. And again, this is now, as everybody knows, a fundamental part of how we're taking care of patients currently. So moving from the tumor sequencing to the germline sequencing, there have been a huge number of studies identifying novel cancer susceptibility syndromes and characterizing the inherited and de novo germline or constitutional alterations that occur in pediatric cancer patients. Common theme across all of them is that when exome sequencing or some similar type of sequencing is used, approximately 8 to 10% of our pediatric patients have a clear pathogenic or likely pathogenic dominant mutation in the cancer susceptibility gene. You can see in the top part of this slide here, some of those genes, P53, APC, BRCA2, NF1, et cetera. And really just emphasizing the point that in addition to thinking about the tumor genomics or precision medicine, looking at the tumor, in parallel, we're equally interested in understanding the germline aspects of these cancers, with the implications that has for the diagnosis of siblings, of other family members, and potential cancer prevention strategies. And this is clearly can be important in terms of cancer surveillance and prevention. This is a paper from a handful of years ago that makes that key point, I think, for patients with P53, germline P53 mutations, Li-Fraumeni syndrome. differential outcomes for patients who either did or did not receive intensive cancer surveillance follow-up and imaging studies, with those with surveillance in the red and those who did not receive that intervention in the blue. Just making the point that although we have pretty limited data for a number of these specific cancer susceptibility syndromes, that from Leaf-Romani syndrome as well as several others, there's data that this actually preventative approach can have an impact for our patients. There's also cases where, in terms of the germline cancer predisposition, other discoveries such as constitutional mismatch repair deficiency, one of the really truly remarkable stories of the last decade, and the survival curve on the left showing the impact of surveillance for patients with those congenital mismatch repair deficiency, with, for example, those who have received full surveillance in the blue, no surveillance in the green. And again, somewhat limited data based on the relatively small numbers of pediatric patients with some of these diseases and disorders, but enough to make the point that, as we would suspect, taking an aggressive surveillance approach for these patients can potentially have an impact. And that might be different, as you can see from the table on the right, might be different depending on which specific gene is altered even in those patients. So key understanding of how genomics can be used to take care of our patients in terms of cancer susceptibility, with the current and ongoing discussion or debate really centering around how broadly that should be used for all patients versus patients with particular diagnoses. So getting back to the tumors, in terms of molecular therapeutics, there's been a number of really excellent examples of cases where genomic studies and doing sequencing to identify alterations in a particular patient can have impact for therapeutic intervention. This study of patients with lymphomas and inflammatory myofibroblastic tumors from a handful of years ago, published in JCO, showing the impact of ALK inhibition in these patients who have ALK alterations. More recently, the story targeting the TRK, NTRK genes, NTRK 1, 2, and 3, with these TRK inhibitors, including in tumor types where treatment in general is really quite challenging. So, for example, in this case, if we're talking about, this is only talking about brain tumors specifically, although TRK inhibitors have been used for many other tumor types. But if you see, for example, in the tumor responses chart on the left, the pediatric high-grade gliomas are those in blue, and adult high-grade gliomas are those in hash, that kind of hash-marked blue. And you can see that there are a number of those patients with those high-grade gliomas that had quite impressive responses. And then over on the right, looking at the various patients, again with the same color scheme, the pediatric and adult high-grade gliomas in blue, patients who had treatment for quite an extended period of time relative to the expected outcome for patients with relapsed tumors of that type. Another example, going back to the mismatch repair deficiency patients, would be the checkpoint inhibitors, PD-1 inhibitors, similar graph to what was shown on the previous slide on the left here, with either using nivolumab or pembrolizumab, that from periods of time that patients can be treated with those therapies for these refractory tumors, if they have hypermutated tumors in the context of germline DNA replication repair deficiency. And here on the left, the CNS tumors are those marked in blue. So again, emphasizing even for tumors that have historically been incredibly hard to treat, there is the potential to see some response to therapies aimed at particular genomic alterations or particular genomic mutational state, in this case, in our patients. Perhaps last, but perhaps not least, for this section on therapeutics, we could have an entire lecture talking about the various trials of MAP kinase, mitogen activated protein kinase signaling pathway inhibitors for our pediatric patients. I'll just highlight a couple of them. This of MEK inhibitor for inoperable plexiform neurofibromas, which are devastating tumors occurring in our patients with neurofibromatosis. showing on the left the progression-free survival curves. So not just survival, but also progression-free survival for patients who are either treated with selumetinib in red versus the expected natural history for these tumors in blue, showing a pretty remarkable difference. And then equally importantly, perhaps showing on the right some of the clinical outcome measures that are seen in patients who are treated with those therapies. So not just that the tumors get smaller, but rather that the patients have improvements in terms of pain, mobility, range of motion, and a variety of the other very important clinical outcome measures. So an example where targeting the MAP kinase pathway can be very effective. Similar pretty remarkable data for patients with histiocytic neoplasms, such as Langerhans cell histiocytosis, progression-free survival curve on the left, quite impressive. And then on the right, showing the outcomes for patients and treatment duration. This is in months, this slide, as opposed to years on one of the last, for patients with different mutations, showing that regardless, in many cases, of the specific alteration in BRAF or some of these other genes, there can be responses in a quite wide range of patients. So again, a pretty remarkable discovery and potential route to treating based on molecular genomic features our pediatric patients. That contrasts with experience for some of these same single agent drugs for patients with perhaps more genomically complex and aggressive tumors, such as this is the example reported from our pediatric match trial, which I'll talk a little bit more about in a few minutes, for selumetinib for patients with relapsed and refractory high-grade tumors of all different histologies, where the progression-free survival here in comparison to that with the more genomically simple neurofibromas or histiocytic neoplasms is really quite different, PFS in terms of several months as opposed to potentially years. And that can maybe be explained likely by the fact that these tumor types here essentially are, and the patients in this cohort, as you can see on the right, are roughly 1/3 of the patients being high-grade gliomas who have been refractory to radiation therapy and other chemotherapy regimens. And rhabdomyosarcomas, also very potentially much more genomically complex, have additional biologic and genetic drivers of those tumors. And example of where the same drug targeting the same pathway for some of the same alterations can have very different effects depending on the actual cancer type. And understanding this better is clearly a large focus of what we're doing. And this shows some of the molecular data for the patients who are treated on that match trial. So again, each column here is a different patient. You can see at the top the variety of different MAP kinase pathway alterations. Notably, a high fraction of these patients with RAS mutations, either KRAS, NRAS, or HRAS mutations, who, even though they occur in the same pathway upstream, have been much less effectively targeted. But then a wide variety of alterations in other genes, and that you can see along the other rows in this chart, even in just this cohort of approximately 20 patients who are treated, another 40 or so patients who are matched to the therapy but did not receive treatment on the trial. So much more genomically complex, and getting a handle on those differences between tumor types is key for us. So I hope in the first half to 2/3 of the lecture made the case that precision medicine is simply pediatric cancer medicine. So there are a number of trials and studies where we're specifically studying genomics or new methods of characterizing tumors. But the fact of the matter is that these aspects, or this aspect of analyzing cancers and our patients, is a fundamental aspect of how we take care of patients in 2023. So there, I'll switch focus to what we've learned a little bit from the pediatric match trial. So the NCI-COG pediatric match is a partnership between the Children's Oncology Group and the NCI, a nationwide screening protocol where patients with refractory cancers of all types, except for leukemia, have their tumors and blood sequenced, and then depending on the results of that testing, potentially can be eligible for one of the match phase 2 trial arms. The eligibility requirements for this trial, which has been open for the last handful of years, are quite broad. It includes patients up to and including 21 years of age. that must have an adequate performance status. And for the first stage of the study, up until 1 year ago, it required a sample being available, a biopsy sample being available at any time of relapse or refractory disease. So not the original tumor that was biopsied or had surgery pretreatment, but a relapse tumor from this patient. And the current iteration of MATCH since January of 2022, outside molecular testing results are being used. So a new fresh tumor biopsy is not required as part of the study. And then the eligibility requirements for the treatment subprotocols are pretty standard for COG or other phase 2 trials. So in addition to having a requirement for the mutation to make them eligible for that trial, they have to have measurable disease and then some relatively standard kind of organ function and washout period criteria. The tumor sequencing for the first 1,200 or so patients on the trial was all performed using this specific DNA and RNA mutation and fusion panel. Samples were processed at Nationwide Children's, and the sequencing was performed at several specific laboratories, including MD Anderson, the MoCA lab at the NCI, and at Dartmouth. So in looking at the patients on the trial, a rough, pretty close approximation of the demographics of pediatric cancer patients in the United States. So slight predominance of male patients. You can see the ages of the patients, of whom it was diverse across kind of the spectrum, but a significant number were kind of in the adolescent and young adult range. And then ethnicity and race corresponding to those, basically matching up the demographics of patients with cancer. It's interesting to see what patients are enrolled on these studies. So this is for the first 1,000 patients, showing about 3/4 of the patients had non-CNS solid tumors, about 25% CNS cancers, and then a very small number enrolled in the trial who had lymphomas and histiocytic disorders. And then looking over on the right, for the non-CNS solid tumors, the key point being made is the high fraction of patients with these aggressive tumors who have various sarcoma types. So in blue, osteosarcoma, orange, rhabdomyosarcoma, gray, other sarcomas, the mustard, Ewing sarcoma. So roughly 3/4 of the non-CNS solid tumors were sarcomas, with a smaller fraction of Wilms tumor, neuroblastoma, and others. So about 75% of 75%, so roughly half the patients enrolled in the study have had sarcomas. For the CNS tumors, we can see some similar expected variability there. About half either had low or high grade astrocytomas or gliomas. Some patients with ependymoma, medulloblastoma, ATRT, et cetera. So a snapshot of the bad cancers that occur in pediatric and young adult patients. So as of the end of the first phase of the trial, pretty much exactly 1 year ago, there were more than 1,300 patients who had been enrolled from across the country, from a number of different study sites. Over the course of the project, it was roughly 25 to 30 patients per month. And the bottom line is, in terms of the ability of the Children's Oncology group and the laboratories involved to conduct a trial of this type, a nationwide screening trial, was a pretty significant success in terms of feasibility. So just emphasizing some of the key bullet points on the left, there had been concern whether there would be relapse tumor samples available for many of our patients, given that until relatively recent history, performing biopsies of relapsed or refractory tumors was not always was not as common as it is today. Of those with tumors submitted, virtually all of them were successfully sequenced, more than 95%, and that small fraction that were not being cases where there really was just not adequate tumor sample available. And then in terms of the feasibility of identifying how often could an actionable mutation, at least as defined by the MATCH trial, which is a very specific definition of actionability. So a patient who had a mutation that made them eligible for one of the 13 MATCH treatment arms, that was found in about 30% of patients. Almost all of those patients were successfully matched to a treatment protocol. And then about half of those patients ended up enrolling in one of the treatment protocols. So a very large scale effort performed by a number of investigators, clinicians, and laboratory folks from across the country over the last 5 years. The factors, one interesting question is whether we could identify factors that were associated with our ability to match the patients to trial and whether they'd enroll on trials. The vast majority of these features here in terms of the sex, age, ethnicity, race, et cetera, were no significant difference seen amongst those different groups. So there was not a remarkably different match rate, for example, for the youngest versus the oldest, or vice versa. A slight increase in terms of patient frequency of actionable alterations found in the younger patients, but not really significantly. But the keys being that there were a few differences detected, as highlighted down at the bottom. Number one being if the patients had previously had molecular testing outside of match. Obviously, if they were at a higher probability of matching to the study, indicating that patients were being selected for enrollment on pediatric match, emphasizing it's not a true survey of all the different refractory cancers, but rather those where the treating clinicians and sites had some reason to believe, based on the tumor type or previous molecular testing, that they might have a match. The treating institution, patients who are treated at sites with a member of the PEP-CTN consortium had a slightly higher frequency of matching, but there was still a significant match rate at all of the sites across COG. And then the CNS solid tumors, as we'd expect from the our understanding of the molecular features of those tumors, had a higher rate of alterations detected. And then of those patients who matched, those who enrolled, the fraction was essentially consistent across all of these groups. So there were not differences in whether a patient would actually enroll in the study based upon their age, race, ethnicity, et cetera. The reasons that patients had a match but didn't enroll on a protocol were, I think, in retrospect, pretty predictable based upon our experience with other early phase trials and an understanding of the challenges for patients and families of making decisions in the time of relapsed or refractory disease. Patients receiving other treatment, so they had the screening performed, but they're receiving some other treatment and potentially still at least temporarily benefiting, so not treated on a trial. Poor clinical status, having an ineligible diagnosis, having received prior treatment with other molecular therapies in some of these categories, for example, off study. And really just, I think, a snapshot of the various challenges of enrolling pediatric patients on early phase clinical trials. I think the other really key accomplishments of MATCH is in addition to providing access to molecular testing for patients with refractory tumors across the country, also providing access to trials of targeted therapies to patients from across the country. So this is the examples. These are the first, actually, these are the 13 drugs that were taken on as part of the MATCH trial. Each of these had a phase 2 protocol. You can see on the right some description of a brief description of the molecular alterations for each. Arm K, ivosidenib, is for patients with IDH1R132 mutations. Arm M, tipifarnib, for those with HRAS mutations. Arm N, sulfacetinib, for those with RET mutations or fusions, for example. A number of these studies opened initially in 2017, and then a few others, as you can see, opened later in the course of MATCH. And a handful of these trials have now been completed, and have been reported and are being reported. Others are still ongoing. This slide shows the fraction of patients or tumors who had an actual mutation for each of the study arms. The key points here, I think, really being that the blue, that's the highest bar, and about 11% of patients were the drugs targeting the MAP kinase pathway, selumetinib, ulixertinib, vemurafenib, and then kind of some of the others are really quite small fraction of patients, such as those in Arm K with IDH1 mutations and Arm N with RET fusions. So we did have an opportunity to see in reality, kind of, we could predict the frequency of these types of alterations, and in general, we're pretty able to do that based upon previous research sequencing studies. Then if you just consider that a tumor profile, that was for the entire cohort, thinking about it for individual cancer types, it obviously varies incredibly. So this is for low-grade gliomas, essentially huge fraction having alterations in MAP kinase pathway genes, a decent number having, as you can see in the orange, FGFR alterations, and extensively, more or less nothing else. where if you look at high-grade gliomas, you still see the MAP kinase alterations and a few FGFR in orange here alterations, but then you see other things such as the gold is PI3K mTOR pathway alterations. So different patterns of mutations in different tumor types. The MAP kinase pathway alterations are incredibly common in several specific tumor types, clearly. So rhabdomyosarcoma, low-grade glioma, and high-grade glioma. But then in terms of patients with all of these different tumors, there's a small fraction of patients with virtually every diagnosed scene that would have some alteration that could be detected in the MAP kinase pathway that would be potentially targetable. And the patterns of the specific alterations, as we know, in different tumor types is different for even just the alterations. So all the tumors that have some alteration in this MAP kinase pathway, as you can see visually here between the all tumors, the non-CNS tumors, the rhabdomyosarcomas, the specific alterations or genes are quite different, which has potentially pretty significant implications for which targeted therapies in targeting those pathways can be used and what diagnostics we might need to use as well. So you can see here for the CNS tumors, for example, the gray are BRAF fusions, the orange are BRAF V600E, which make up a very large fraction of alterations, both in low and high-grade gliomas, and then a scattering of other tumor types. So I think this one of the things the study emphasized was, relatively speaking, the very high fraction of patients with a variety of pediatric and young adult cancers who have alterations in MAP kinase signaling. And we knew this, of course, but it was a very stark reminder of that. The arms for the MEK inhibitor and ERK inhibitor were, the two first to be completed as part of the study. And there's continuing emphasis, including an upcoming arm of the NCI combo match trial targeting this pathway, to really continue to study combinations of therapies, not just single agents targeting MAP kinase signaling. So that will be the next step. As part of the match, we also had an opportunity to see a snapshot of the tumor alterations that were detected and the diversity of the genes that we saw there for different cancer types. I won't belabor this, but these data are all available as part of this paper that was published in JCO this year. But I think the one key thing to emphasize is what we would call optimistically opportunities for continued discovery, or more pessimistically, some of the key challenges for therapeutics in pediatric cancers. The gray bars are ones where there were recurrent alterations that were detected in a gene that was not a focus of an arm of match. So P53, MYC, beta-catenin, MYC-N, the histone gene alterations, et cetera. And just emphasizing that in pediatric cancer in particular, there is a wide variety of these alterations that really, there's ongoing very exciting work focusing on each of the biology of many of these genes, all of these genes and pathways. But in terms of currently available therapies, kind of, and the front of investigational therapeutics, it's much more sparse. So in conclusion, before we get to questions, I'd say that we've really had remarkable progress over the last 2 decades. So from about early 2000s to now, gone through the invention of large-scale sequencing, technological and bioinformatic developments to allow analysis of large-scale sequencing, using that to better understand a wide variety of both pediatric and adult cancers in terms of their underlying biology and genetics, and then applying them, and they're both the results of those tests as well as the methods and technologies themselves have been incorporated into the diagnosis, prognosis, and therapeutic decision-making for our patients, in addition to better understanding cancer susceptibility and guiding potential prevention strategies. So precision medicine, cancer medicine has become, as I said a couple of times now, just simply one aspect of pediatric cancer medicine. The MATCH trial, I think, is an example of how a number of things, but one being how effective collaborative precision medicine trials can be if we're looking for alterations that are in very specific and very small numbers of patients. Nationwide screening and sequencing trials are potentially a very useful way of doing that, and also, very importantly, facilitating patient and family access to those testing and trials. And through initiatives such as the NCI's Molecular Characterization Initiative, through COG and others, these type of sequencing that we're performing as part of MATCH and more extensive sequencing is now being done both in part of clinical care and as part of trials pretty routinely for our patients. And again, the data, including those of the specific genes that are found to be altered, as well as the early results of some of these trials, emphasize the need for continued innovation and even more broad clinical translation for our patients. I'll end by just highlighting one of our patients who we've taken care of here at Texas Children's, my colleague, Dr. Lynn, and others. The 6-year-old boy diagnosed with an anaplastic ependymoma and histology, not shown here, but absolutely classic for anaplastic ependymoma. One of the bad versions of ependymoma where tumor progressed rapidly after radiation therapy, spread throughout the brain and spine. We had no additional treatment options. Genomic sequencing revealed an NTRK fusion, which had never previously been reported in ependymoma, and I think emphasized the potential difference between what we see under the microscope and what we can detect by molecular profiling. And it's one that potentially be targetable with NTRK inhibitors. And this is a patient now who's been successfully treated with larotrectinib for more than 2 years, coming up on 3 years, I believe, and had really minimal toxicities of that therapy. And I actually live currently in an apartment on the Rice University campus, and I somewhat frequently see this guy coming around. This is a slide of him, photo of him at one of the Rice basketball games, hanging out with his new best buddies, the Rice cheerleaders. So really remarkable progress for individual patients, and the hope is that we can make this even more broad. We're thankful to all our patients and their families participating in the studies. I won't go through the entire even small version of the acknowledgements of those who are involved in pediatric MATCH, other than to say that Nita Seibel is the NCI study chair, Katie Janeway is the vice chair from COG. And you can see in the names in the column on the fourth column here, for each of the treatment arms, there are also individual investigators from COG who are involved, both as the study chair and vice chair. And this is really all of their work that I'm giving an overview of here today. So with that, I think I will stop sharing my slides, and I'm happy to take questions at any time that we have left.
Wow. You packed a lot of information into your presentation. Before we go to the questions that have been submitted, I had one or two that I wrote down. So I was trying to follow along, and when you got to the breakdown of the types of tumors that you guys have seen, it seemed like osteosarcoma may have been it seemed like 25% of the non-CNS tumors were osteo.
Yep.
And was that high for osteosarcoma? And if so, did you see like more tumors coming in from some groups of doctors than others?
So that's a good question. So in terms of where patients enrolled, we did not see really marked patterns of coming from particular places. There were some individual hospitals that had other screening, pretty rare, that had other screening tests or other screening things that would not participate as commonly as you might expect they would by the number of patients. But in general, there were, I think, patients enrolled from now more than 140 different hospitals across the COG, and only the US part of COG was restricted to for the trial. In terms of osteosarcoma specifically, I think it just emphasizes, so that's a larger fraction of patients than if you're looking, you usually see the pie charts not of refractory or tumors that haven't responded to treatment, but rather the pie chart of newly diagnosed patients. And that's clearly far more patients than you would expect for osteosarcoma at diagnosis. But I think what it really emphasizes is that sarcomas in general and osteosarcoma in particular make up a pretty high fraction, a disproportionate fraction of our patients who have not responded well to treatment, right? We know there's kind of the progress of pediatric oncology over the last 10, 20, 50 years has been becoming quite successful, at least in terms of survival outcomes at treating a wide variety of cancers, from acute leukemias to other tumor types. But really now what we're left with are a subset of cancers, such as the high risk and metastatic sarcomas, such as some of the very bad neuroblastomas or Wilms tumors, some of the high grade gliomas, and others that are kind of the outliers, right, that are still the remaining challenge. So sarcomas in broadly in osteosarcoma in particular are desperately in need of new therapeutic options. And this trial was helpful in that we could find molecular alterations in those patients, but even though osteosarcoma, for example, is really complex in terms of its genome, there are not a lot of kind of classically targetable things there using the kind of drugs that I've been talking about. So immunotherapy approaches and a wide variety of other investigational therapies are being looked at for that.
So I guess it was a sign of need where they may not have had anywhere else to go.
I think that's absolutely right.
If anybody, if anybody has questions, you can click the Q&A button at the bottom of the screen and submit a question. The next question is was submitted was what drove the choice of drugs on the match trial? And the example they gave was why ensartinib over lorlatinib.
Yep. So two things. One was we wanted to get drugs targeting the common genes and pathways that were altered in pediatric cancer, right? So we wanted something targeting BRAF and NTRK and, you know, the things that we knew would be common. But after that, it was largely questions of timing and availability from the drug companies. So if we had for example, if we were looking for drugs targeting a particular gene, we, through our NCI colleagues, approached all of the different companies who had drugs kind of in that space. And some of them, either because of timing or because of other trials they're running, were more enthusiastic than others about participating in the trial. I will say in general, we were very lucky to have such great partnership from the pharmaceutical industry. That the 13 arms of Pediatric Match represent 12 different pharmaceutical companies, all collaborating as part of the study. So the short answer is what we thought were high impact genes and targets in pediatric cancer, and then feasibility, essentially.
Thank you. I might have missed this at the beginning because I had to step away from my desk for a minute, but is this continuing now, or are you guys, did you stop it for a while?
So it hasn't stopped. It's transitioned into a new phase. So from when it opened in 2017 up until the end of the end of 2021. So a year ago was the phase where all the testing was being done centrally. So samples had to be available. A patient could not be enrolled if there was not any tumor available to send to COG. And at one of the central labs, we would do the tumor sequencing. And that was the plan that was approved by the FDA and the NCI and all of our partners and the appropriate regulatory colleagues. Starting then at the beginning of 2022, we moved to the trial still open, and the arms that are still that haven't yet been completed are still open, such as a handful of the ones I emphasized. But we're now using outside molecular testing results. So it's the testing for patients has become increasingly common, and there is actually a way to get testing at no cost through a particular company as part of the trial. But the landscape had really changed between when we were planning the study in 2015 and 2022. And one of our concerns was always that, we felt like in the beginning of the trial, in the first phase of the trial, we needed to have a tumor because we needed to sequence it ourselves and we needed to see what was potentially there. But doing that type of testing has become sufficiently available for patients across the country now that in the interest of not limiting patients from the study who don't have any tumor left available for a trial, we started using outside results. So it's still ongoing. It's a much reduced rate at this point, right? In the peak of Match, we actually were enrolling patients at 40, almost 50 patients a month. The average for the study was more like 25 to 30. But now that we only have a handful of treatment arms open, and they're by definition some of the more rare treatment arms, right? So all of those for the BRAF, the MEK inhibitors, the ERK inhibitors, those MAP kinase pathway inhibitors, which are the most frequently altered genes, those have been completed and are being analyzed and reported. So we're now left with some of the true, more rare things, such as the HRAS mutations, the IDH mutations, and others. So short answer, study's ongoing. It's a remaining handful of arms, and it's done exclusively using outside molecular testing results. And if anyone ever has a question about that or feasibility or about access to testing for a patient, they should please just reach out to me, and I'd be happy to help them figure it out.
So the decision whether to reopen other arms will come after you guys have a chance to analyze outcomes?
Yeah, so that's a good question. So there will not be new treatment arms as part of pediatric MASH. Like this particular mechanism will be ending when, sometime in the future. But the interesting thing now is like, so there's other mechanisms. So there's now the NCI is their combo match kind of trial, which is equivalent to match, not just for kids, but also for adults, that this combination therapies. And so we have the opportunity to put forward pediatric trials as part of that study. And one has already been put forward targeting MAP kinase signaling that hopefully will open within the next year. But then also through other trials, through, for example, the COG, PEP-CTN, and other mechanisms, these sort of treatment arms that we're talking about for pediatric MASH are now done more routinely as trials of their own at this point, right? I mean, it's again, an interesting and predictable and logical evolution of kind of state of the art of molecular testing. Now, screening a patient's tumor and enrolling them based upon some molecular eligibility criteria on a clinical trial is kind of standard operating procedure for us in COG in a number of trials and through all these different consortia. So I think the need for a nationwide screening trial such as MASH to ensure testing and access for patients is much reduced, which is why kind of we're moving on to a different model. And it's all also, of course, about it's been a very, a lot of investment from the NCI in the trial and now in other initiatives as well. So MASH at some point will be coming to an end.
Okay. So if you had a family who had a child who would possibly be eligible for one of the arms that's still open, but
Yep.
they had a treating physician who was not open to investigating it, is there a way that they can talk to somebody, just in case we have families that are listening here?
Yes, the most straightforward way is to reach out to me directly.
Okay.
My email and phone are easily Googleable.
Okay.
You're welcome to share it, obviously, with any families, but
Okay. Thank you.
It's very straightforward for us to help people figure this out.
Okay. Another question that came in says, do you envision a time when there'll be a molecular diagnostic test like they have for breast cancer to evolve for the different types of pediatric cancers?
I think there will be. So that test for breast cancer is basically kind of classifies based on gene expression risk and kind of puts patients into different categories of risks that helps kind of give prognosis and also assign treatments. So for some cancer types, such as medulloblastoma, for example, that's what we're already doing, right? I mean, it's a different test, but practically speaking, patients are being categorized in the same way. I think over the next handful of years, for additional cancer types, we'll have equivalent tests like that as we have a better understanding. In part, we have one disadvantage from our adult colleagues, which is if someone wants to study a test such as that for breast cancer, for example, and its clinical features, they can study thousands of patients. And I mean, it takes work, but it's feasible to do it. Whereas for many of our diagnoses, we're talking, 100 patients a year, 500 patients a year for some of the big ones, 20 patients a year for some of the others. So getting kind of the robust clinical data so that we can say for sure what a particular molecular profile means is more challenging. But I think that'll become more and more common. It's part of what we need to be doing.
Well, thank you, Will. That is the last question, and which is good because we are out of time. But I want to thank you so much for your presentation. I've heard so much about the match over the years as you were getting ready to launch it, and now I can't believe it's been 5 years.
Yeah, it's been open 5 years. We're planning a few years before that, so it covered a long part of my career, certainly.
But I feel like I just got like the 101 version of being brought up to speed about what you guys were doing and how you did it. So I really appreciate that, and appreciate you taking the time and everything you have done for Alex's over the years, Will. So thank you, and we appreciate you.
Thank you. I'm very grateful for your support, as are our patients and their families.